embryonic stem cells, induced pluripotent stem cells, or adult stem cells. So we have discussed that with the help of these kind of stem cells, we can cures, we can study the disease models, or we can study the impact of drug, or we can study various kind of therapies to cure, or to um, to cure some or the other diseases. So till now, can anyone brief me how many models we have discussed till now? Can anyone brief me? Old students which have who have attended all the sessions till now, they must be aware. Anyone want to answer? No? Okay, so till now we have discussed about the cell lines, or we have discussed about the cells, only stem cells, which can be differentiated into a particular organ, or into a particular sorry, organ, not organ, or to a particular tissue, and on which we have tried to study some or the other drug discovery, or some or the other therapies. Today we'll discuss about the organoids. Organoids are the 3D structure where we can study about these drug responses of these drug responses of the anything we engineered in lab or something in detail in a 3D structure. So organoids are three-dimensional miniature version of organs or tissues that are derived from cells with stem potential, such as embryonic stem cells, induced pluripotent stem cells, or adult stem cells. These structures self-organize and differentiate into various cell types, and they mimic the key functional, structural, and biological complexity of the corresponding organ in vivo. We have discussed about in vivo, ex vivo, and in vitro experiments.
Yes, ma'am, we have discussed about.
Yes. Okay. So these 3D structures, till now what we have done, we have discussed about the 2D models for our drug discovery and for our this tissue engineering. Okay. So can you tell me which of the 2D structures, 2D models we have studied till now? Anyone? Try to be interactive in this session, please. No one is answering except one. What happened to Vinod and Nidhi, and one more is there? Okay, so basically, till now we have discussed that on a plate we grow some cells, and some or the cancerous cell, or some or the non-disease cells for our study. But in this, what we do, we create a 3D model in our labs, and we study all these all the impact of the drug which we are formulating in our lab for some or the other treatment, or any other kind of some or the other therapies which we are experimenting in our lab, to study them extensively on a 3D structure. So these organoids are used in that kind of things. So using stem cell, we can design a 3D structure of brain, a 3D structure of a heart, 3D structure of your kidney, and so on. So like this, we can target each and every portion of your organ to cure some or the other disease, to employ therapies which we are studying in our which we are studying in our labs for these treatments. So these organoids are used in research for studying development and disease, drug discovery, personalized medicine, and tissue engineering. So please let me know if my slide is changing or not. Is my slide changed?
No, ma'am. Yes.
No or yes? I
It
heard
changed.
change. Okay. Are we on third slide? Engineering the 3D structure and cellular organization of organoids.
Yes, ma'am. We
Yes,
are in the
ma'am.
third slide.
Okay, okay, thank you. So now we'll discuss about some of the key characteristics of the organoids. It is three-dimensional structure, as I have already mentioned. Organoids are grown in a 3D environment, which allows them to mimic the spectacle organization of tissues in the body. So basically, what we'll do, we'll create a 3D structure of a particular cell or particular tissue or a particular organ in a lab, and we provide it a similar environment as they got in our body. Okay, so we'll exactly mimic the things which are happening in our body in our labs. So this model or this thing is called as organoids. So this allow them to mimic this allow these 3D structure of a organ or tissue, you can say, as they are mimicking in our body. So these three dimensional structure of organoids is a critical aspect of their ability to mimic the complex architecture and functionality of human organs. So they have some key features like self organization. So do you know what is the meaning of self organization in for our tissue or organ in our body? Anyone?
Ma'am, can you repeat the question again?
The question is, basically the first key feature of the 3D structure of these organoids is that they are self organized, or you can say self organization is the main characteristics of these 3D structure. So what do you understand by the term self organization? You can try to give the answer.
Ma'am, may I?
Yeah, sure.
I mean, is it like the ability of those cells, to arrange them into complex structures without, external applications or by their own?
Yeah, as our organ, as our tissue, they self organize themselves into the in our body environment. Do you supply anything to grow them, or do you supply anything to make them heart, or to make them a brain or kidney? No, we do not provide any particular thing to them. They organize themselves into a particular system, into a particular organ, into a particular tissue, um, in our body environment. Exactly. Okay, so like this, these organites also self-organize in themselves, and the self-organization in the context of organites refers to the process by which cells, often derived from stem cells, interact locally to form higher order structure and patterns without external direction. Okay, as one of you mentioned, we do not apply anything externally on them. We just provide them the environment which is similar to the environment they got into a body, and they self organize themselves to be a brain, to be a heart, to be a kidney. Okay, this process is crucial for organoids to mimic the morphology and function of tissues, or you can say organs. So till now we haven't studied about the tissue and the organs model in our lab. We have just studied about the particular cell. Okay, a cell of a brain, cell of a heart, cell of a kidney, cell of a cancer patient, cell of something, cell of something. Okay, so today we'll discuss about these organoids. Basically these are the tissue or the organ. Okay, so specifically we are dealing with a proper structure and a proper model of a proper model of this organ present in your body. So is this difference clear to all of you, that what is the 3D structure and what is the 2D structure we are discussing here? Yes, no, any hand raise or hands down, anything? Or should
Yes,
I
yes. Can you
repeat?
repeat the question again? Can you repeat the question?
is the difference between the 2D model, which we have already discussed about these disease modeling and some therapies, and the 3D model which we are discussing today. This difference is clear, or you want me to repeat one more time?
Like, can we have a slight revision of just the
Slight,
difference?
yeah, sure. I don't have any problem. Okay. So basically, till now, we were discussing about a particular cell which we are extracting from particular organ. Like we are dealing with the brain cell, we are dealing with the heart cell, we are dealing with the kidney cell, or we are studying the cancerous cell. We are discussing upon these cells which we grow in a plate or in a flask with a particular media. Okay, so till now we were dealing with the 2D structure for the disease modeling or for the discovery of any particular drug or compound. Today, we are discussing about the thing called as organoids. Organoids are the 3D structure of any particular tissue and organ present in our body. What we are doing, we are designing this particular organ or tissue in our lab, and this is a whole organ. Uh, it's a 3D model of whole organ on which we are studying our drug, which we are formulating in lab for some treatment, or on which we are studying a particular treatment for some for the cure of some disease. So these organoids are the 3D structures, 3D structures that mimic the organ which are present into our body. We particularly provide an environment provide the environment which is similar to the environment of our body in the labs to these tissue and organs, so that their functionality, their morphology is exactly similar to that which we have in our body. So these are the organoids. Is it clear now?
Yes, ma'am.
Okay. So for only one student or two others also?
Yes, ma'am.
Okay. Yeah. Thank you so
Ma'am,
much. Yeah. So these structures help us to target each and every corner of the organ, because you know that if we have something related to a brain, if we are dealing with some or the other tumor which is in the brain, or some other disease which is in the brain, so we can't say that it is in a particular portion. Oh, sorry, we can't say it that we have to treat the whole brain. No, sometimes the disease is target specific, and in 2D model it is very difficult to target that particular portion in the brain. So in 3D structure we can easily find out the main target where the disease is causing, or from where the disease is causing. So we can just target that point in the brain, and we can treat it with the help of some or the other drugs, which we can treat it with some or the other therapies. So the advantage of these 3D model is that we can target a particular site, or we can target a particular portion where the problem is occurring. Yeah, someone wants to say something. Okay. So we were discussing about the self organization. Yeah, in our body we do not provide anything externally for the for the morphology or the for functioning of particular tissue or a particular organ. Exactly. Am I right? If we are talking about the development of brain, or if we are talking about the development of our any organ present in our body, do we provide any supplement without any disease or something? Do we provide anything to them? I'm asking
No, ma'am.
something. No, yeah, we do not provide anything for the development. We treat them for their betterment or for some disease. We do not provide anything for their organization, for their development, or for their morphology, or for their functionality. They just do it by themselves as we grow from a child to an adult, or so on. So similarly, these organs also, the key feature of these 3D structure is also self-organization. In the context of organoids, refer to the process by which cell, often derived from stem cells, interact locally to form higher order structures and patterns without external direction. We do not give them any direction. We just provide them the environment which is similar to the body. Okay. This process is crucial for organoids to mimic the morphology and function of tissues or organ. So, these some key features of self-organization is local interaction. That is, self-organization is driven by local interaction among cells, which includes cell-cell and cell environment interaction. Do you understand what is the meaning of cell-cell and cell environment interaction? Do you know that? Okay, so you all must be aware that tissue is made up of multiple cells from 1, 2, 3, 4, and millions of cell. One tissue is formed, and from tissue organ is formed. Am I correct?
Yes, ma'am.
Yes.
Yes, ma'am.
Yeah. Similarly, local interaction means we take some cells, similar cells, and we provide the environment. Now, these cells will interact with each other to form a tissue and grow in a particular tissue or in a particular organ because of the cell environment interaction. Basically, multiple cells are interacting with each other to form a tissue, and this the functionality and the morphology of this tissue is promoted by the environment which is provided by us in the in the laboratory to these tissue. So this local interaction is self organization is driven by local interaction among cells, which includes cell to cell interaction and cell environment interactions. These interactions involve intrinsic and extrinsic cues such as some type of gradients, extracellular metrics, and mechanical properties. Okay, so basically, um, the how these cells combine together to form a tissue in our body. Similarly, this is also happening in the laboratory, and these cell to cell interaction forming a particular tissue, and the morphology and the functionality of these tissues are given by the environment which we are providing in the lab. Then next is the emergence of patterns. So through these interaction, patterns emerge at a global level, leading to the formation of complex tissue structure, um, such as villi in intestinal organoids, or the epidermal layer in skin organoids. So how the, you know, from the single cell a multicellular organism form, and all these cells develop themselves into a particular kind of cell or a particular kind of organ, and all the organs have different properties and different morphology. Correct?
Yes.
Similarly, after the cell to cell interaction and after the tissue formation, these cells form some kind of patterns. Like in intestinal cells, we all know that for the movement of food, we need villi, and so this is the kind of development in this particular cell. So intestinal organoids develop villi inside them, and we all know that in skin cell we have a layer of uh, epidermal tissue. So like that, in case of skin organoids, you can say the development of epidermal layer self organized itself. Okay.
Yes.
uh, do you have any doubt till here? You want me to repeat anything? I can do that for you. Or should I
No
continue?
doubts, ma'am.
Okay, thank you so much. So, next key feature is the distributed control. The process is not controlled by a single cell or external factor, but is distributed among all the components of the system, making it robust to capable of self repair. You all are aware of self repair. We are discussing it from class 1, that all cells have the capability of self renewing themselves or self repairing themselves. So the similar properties also present in these kind of organoids also. Okay. So now moving on to the importance of self-organization. Can anyone tell me what is the importance of the self-organization? Why do we need this kind of thing in these organoids? Anyone?
Because once we
Ma'am,
are
can you
given
repeat
an
the question?
environment,
One by one. You can raise your hand and I can call your name.
yeah, ma'am. Because
Yeah, please,
once we are
Sunil.
giving, yeah, once we are giving a micro environment in the lab, and we expect
Yes.
a typical kind of a tissue to grow, we want
Hmm.
it to be self-replicating and not from some other kind of tissue which can cross over, have a rather fatal outcome. Like if we are giving the environment to grow cardiomyocytes, then we are not expecting them to convert into any other
Correct.
kind of allied tissue. That is what is our intention.
Yeah, basically, if we're talking about a micro environment, we are particularly giving, if we want our cell to be a heart, or if we want to be our cell to be a kidney, so we provide this similar environment in which a heart cell can grow or heart tissue can form. And if we want to grow it in a skin type of cell, we will give that kind of micro environment to that cell. So this is the importance of cell organization. And if this is not happening by its own, then we will face some or the other problem when we incorporate it into a in when we deal with some or the other therapies with it. Suppose we are providing micro environment of kidney to a heart cell, and now we are doing some or the other therapies to treat a cancer related to skin. Will it work on this kind of tissue? No, it will not. So we have to make it very clear that if we are dealing with a brain cell, or if we are dealing with the brain cell, we have to provide it a particular environment. Or if we are dealing with the kidney cell, we have to give it a particular environment. That's why they self organize themselves in accordingly. Okay, so the importance of cell organization is tissue morphogenesis, as Sunil has already tell us discussed with us. Self organization is essential for the morphogenesis of tissues within organoids, allowing them to um, recapitulate organ like structure and function. Sunil, you can mute yourself, please. And the regeneration and repair, for the regeneration and repair, this process enables organoids to regenerate and repair tissues, which is valuable for therapeutic applications. Okay, so this is the importance of self organization. Now we'll come to the complexity and organization of these organoids. Wait, let me change the slide. Is my slide changed?
Yes, ma'am. The slide is changed.
Okay, so now we'll discuss about the complexity and organization. So organoids represent the complexity, organization, and structure of tissues by mimic the intricate architecture and functional interactions found in living tissues. This is achieved through their three-dimensional structure, which allows for a more accurate representation of in vivo cell responses and interactions compared to the traditional two-dimensional cell structures. I have already discussed with you that in if we are testing our drug on a 2D model, we are just targeting a particular kind of cancer or particular kind of thing. Okay, we are not dealing with the whole organ or a whole tissue. You uh, uh, as we have also discussed about the if we are treating one cell, and in treatment of that particular cell we are affecting the nearby cell. So in this was the limitation of that kind of 2D modeling. But in case of this 3D structure, what we are doing, we are treating whole of the organ, and all the cells are in our control. And if a disease cell is basically, if we target a disease cell, we can also treat nearby cells, and we can also cure the spreading of that infection, or spreading of the negative impact on the nearby cells. So yeah, this kind of organoid cells reduce the complexity of the these kind of therapies. So this is achieved through their three-dimensional structure, which allow for a more accurate representation of in vivo cell responses and interactions compared to the traditional two-dimensional cell cultures. So the key aspects of how organoids achieve this complexity and organization, this is the organ the first one is organoids are composed of multiple cell type organized in a 3D space, which facilitates complex cell to cell interaction, signaling pathway, and mechanical forces that are crucial for tissue function. Okay, so what you get it by the thing signaling pathway and mechanical forces that are crucial for tissue function. Can anyone tell me how these things are solving our problem in case of 3D structure and not in 2D structure? Anyone?
Yeah, ma'am. Can
Yeah, please. Yeah, sure.
I, every tissue in the body has a different function too. Like some are secretory organs, some are having peristaltic movement, some are having intrinsic activity like cardiomyocyte. So when we are
Correct.
evaluating in a 3D model, we are also seeing the effect of the drug or the targeted therapy of how they will function or how it will alter the function of a normal tissue in the body. So that is what will give us an interference of the targeted approach toward that particular tissue. Like if we are testing for a drug which can increase peristalsis in the intestinal tissue, so we require a 3D model of intestine to test on.
Yeah, correct.
And and also if you want to know that how cardiac myocytes can be made to move faster in case of cardiac failure. So if a targeted drug is to be tested, we need a 3D model of cardiomyocyte. Similar approaches like
Yeah,
in
same.
pancreatic tissue. So if you want to see the effect of a typical drug which will enhance the production of insulin, so we need a 3D model of pancreas with those
Pancreas.
typical beta pancreatic cells. So that is how
Correct.
the tissue 3D model
We are
generation will help.
Yeah, if we are just dealing with a single cell, we do not know what will be the impact on the whole organ of that particular drug or a particular therapy which we are giving to a patient. Okay, so if we are talking about signaling pathway, you all must be aware that signaling pathway is not a work of only one cell. It is a connection of organs, in fact, from one organ to another organ to another organ. So we need to study, if we need to study all the pros and cons of these kind of therapies, or these kind of drugs which we are using or which we are designing in our lab, we need to thoroughly thoroughly study the impact of these kind of things on the particular cell, particular tissue, or particular organ, as well as to the nearby cells and organs as well. So yeah, that's why 3D dimensional models are way much um, what we can say, important as compared to the 2D structure. Now, if we say multicellular composition, they contain various cell types, including epithelial, stromal, and sometimes vascular or neural cells, which contribute to their functional complexity. Okay, so this is the multicellular composition effect on the complexity and organization. Next, if we discuss about the advantages over 2D structures or cultures, which we have discussed in our previous lecture. So the first one is realistic cell interaction. Can anyone tell me what do you mean by realistic cell interaction? We have already discussed it, but I want to know how much aware you all are. Yeah, some other person now I want to discuss this with. No, Sunil, you will not answer this again. Anyone else? Or only Sunil is attentive in this class?
Ma'am, can you please repeat the question?
Yeah, sure. So I was asking, if we are talking about the advantages over 2D cultures for 3D, so what do you mean by realistic cell interaction? What you are seeing in 2D cultures, or what you are seeing in 3D cultures or 3D structures, what is the realistic cell interaction? We have already discussed it. So Nidhi, please try to answer.
Ma'am, it is through signal uh, interaction, through signal interaction.
No, I'm talking about the cell to cell interaction, or how cell are interacting to the environment or to the nearby cells and other things. We have 22 attendees in this session, and only
Ma'am,
one is. Yeah, your name,
can I
please.
answer? My
Yeah,
name is
sure.
Rujan.
Yeah, please, please.
Is like a realistic cell interaction. I mean, is it like mimicking natural development of the tissue function, and
Correct.
like they have interaction based on biochemical pathways and
Exactly.
electric.
So here, what we are discussing, that if suppose you have a brain tissue, and we have incorporate some or the other kind of tumor to study how to deal with this tumor, or how our therapy is going to treat this tumor in our brain. So we have a structure, we have a 3D structure of our brain. Okay, now we have induced a tumor in that uh, brain, and now we want to study the therapies or the drugs which we are developing in our lab. So in case of realistic cell interaction, what is happening, the disease progression will be real. Okay, you cannot say that in brain has four portions, and we cannot say them that every time it will be on the same portion, the tumor will be on the same portion. It may be in some or the other portion, or it may be in more than two or more than one portion as well. So, or it may spread from one portion to another portion. So we have to make all the cases, uh, we have to treat, or we have to be ready for each and every kind of kind of tumor which can be or which is happening in the brain. So the disease progression is means how disease is happening in the brain, or how disease is happening in some or the other organ, will be real in these kind of structures. So the treatment also, if now we are treating such kind of cancer which we have induced in this kind of model of the brain, we can treat it accordingly. We can treat each and every cell of the brain which we have in this 3D structure. Okay, are you getting my point, what I'm trying to convey through this realistic cell interaction, or should I repeat?
Ma'am, doubt.
No, ma'am.
Sorry, Saloni,
Doubt. I
you were
have a
saying something?
doubt.
Yeah, please.
Ma'am,
You can write your doubt because your voice is not clear to me. You can write in chat box and I will answer here.
Okay, ma'am.
Yeah. Or you can try once again, maybe. So, in meanwhile, yeah, I got her question. Network issue, sorry. What is the question? You are saying network issue. Sorry. What is your question, Saloni? You have a doubt now? Okay. So, if you, I was asking, like, how can we know the further complication or reaction of the treatment? Yeah, basically, that is we are discussing over here, Saloni, that if you have a tissue of a, when you have a brain organoid, okay, now because we are dealing with one particular organ, yeah, if you want to have if you want to study about the impact of treatment of brain cancer on your other other organoids or other organs, you can do that also with the help of creating with the by creating the microenvironment for both the organs, and uh, by by connecting the signaling pathway, common signaling pathway between them. Okay, because no, all pathways are not interconnected, but yeah, from you can say from 100 to 80 are connected, so you can create that environment in your lab, and that according to that you can design your organoids, and then you can study impact of that treatment on the other organ or on the other kind of tissue present in your body, or other kind of cells in on which you want to study, or on which you want to see the impact. Okay, so basically the signaling pathway can help you to to sort out things between like if you are treating brain cancer, so your neurons are connected, so your spine, you can check the impact on the spinal cord, if because these are the two majorly connected things, so you can check the impact of that treatment on your spinal cord. Okay, so you can work on that signal pathway and create your micro environment according to that. Okay, so are you getting, Saloni? Your doubt is clear now? Okay, ma'am, thank you. Yeah, sure. So again, the another advantage is improved drug testing. This is also we have discussed, the complex structure of organoids make them more suitable for drug testing and toxicity study, providing more accurate prediction of drug effects in humans compared to 2D models. Okay, so we can improve our drug testing thing with the help of these structure. Now coming to the mimicry of organ functionality. Uh, you all must be okay if I'll continue this lecture till 6:15.
Yes, ma'am. We are okay.
Yeah, yeah, because session was we have started it late, so that's why. Yeah, now we'll discuss about the mimicry of organ functionality. Organoids can perform specific function of the organ they model, such as contraction or secretion, due to their 3D structure. As we as I don't, I think Sunil have answered that. Like some organs are involved in secretion, some are doing something else, some as so basically in this organoids the functionality is defined. These organs are very much defined, how they have to act, how they have to mimic accordingly. Okay, so organoids are capable of mimicking specific function of the organs they model due to their three-dimensional structure, which allows for complex cell-cell interaction we have discussed, and tissue organization. This functionality is crucial for studying organ development, disease modeling, drug testing. So now, one by one, we'll discuss about one or the other organoids, like intestinal organoids. These organoids can form a crypt-like structure and really like, projections similar to the small intestine, and exhibit barrier function, nutrient absorption, and secretion of digestive enzymes. They can also respond to drugs and pathogens, making them useful for studying gastrointestinal diseases. Okay, So this is how we can study each and everything about small intestine by creating these kind of organoids in our lab. They are used extensively in research to study intestinal development, disease modeling, and drug testing. So the key features again, cryptic villus structure. So these kind of cells, you know using the microenvironment, these kind of cells replicate the cryptic villus architecture of the small intestine with a uh, with a central lumen surrounded by a single layer of polarized epithelial cells. These structures include intestinal stem cells at the base of the cryptis, similar to the in vivo conditions. Okay, so this is this help us to study each and everything about the small intestinal functionality, morphology, drug responses, and kind of things. Organoids contain various cell types found in the intestine, such as enterocytes, goblet cells, Paneth cells, enteroendocrine cells, which are essential for nutrient absorption, mucin production, antimicrobial defense, and hormone secretion. So each and everything is present in this kind of model. Again, self renewal and differentiation. The presence of intestinal stem cells allow organoids to self renewals and differentiate into different cell lineages, maintaining the epithelial layers, continuous turnovers, everything. Okay, so if we talk about the applications, the applications are again your disease modeling. If we want to uh, if we want to study about the inflammatory bowel movement disease, or something improper happening in our intestine, we can just study on these 3D models of small intestine. Or if we want to design some or the other medicine for um, the problems happening in the intestine, so we can test that we can design that regenerative medicine, and we can test it on these 3D models, and we can um, directly send it for the phase 1 and phase 2 trial. Okay, so these kind of things which we can do with the help of these small intestinal organoids. Next, we'll come to the cerebral organoids, which we have continuously discussing, that is brain cells. So these organoids self-organize into neural networks, displaying, cortical layering with distinct neuronal population arranged in characteristic laminar patterns. They exhibit spontaneous electrical activity resembling patterns seen in the developing brain. This makes them valuable for studying neurological disorder and brain development. So these are the 3D, 3D structures of brain cerebral organoids. So these structures can be derived from human pluripotent stem cell, such as embryonic stem cell or induced pluripotent stem cells. These organoids self-assemble to form an organized architecture composed of various cell type, including progenitor cells, neurons, and mimic aspects of brain development and structures. Okay, so in both the cases, like in intestinal case and in brain case, you have seen so much difference in the uh, morphology, correct? We have lots of difference in the morphology of intestinal cells and in case of brain cells. So the microenvironment which we providing to these kind of pluripotent stem cells or induced pluripotent cells are very much different to develop them into a brain or to development into a small intestine, correct? So in case of if we talk about the structure and function of these kind of cell, there are neural zones in these kind of cells. Cerebral organoids contain distinct neural progenitor zones, such as the ventricular zone and the outer subventricular zone, which are similar to those found in the developing brain. Then we have cortical layering. Organoids exhibit cortical layering with early born neurons forming deep layers and late born neurons forming upper layer, reflecting an inside out pattern of neurogenesis. Okay, then the development of the neuronal activity. The neurons in cerebral organoids are functional, capable of action potential and exhibiting spontaneous uh, s- um, activity, although they are less mature compared to the adult neurons, but their activity is like brain only. Again, if we talk about the applications, then disease modeling. Again, disease modeling, brain development study, we can study about how the brain development happening. They provide a valuable tool for understanding brain development, including the formation of neural layers as well, and the maturation of neurons. If we want to see how a brain develop from a newborn to adult, how we can study that stages also using this kind of 3D models. Or you can also study about the blood brain barriers, how the circulation of blood occurs in brain, how all these signals work in the brain. You can study anything related to brain with the help of these 3D structures. So yeah, these are the organoids. Tomorrow we'll continue discussing about these organoids for other organs also. So today we'll stop our class here. If you have any question, you can ask me. You have 2 minutes to that for that. Anyone?
Ma'am, now all these organoids we are talking about are derived from the embryonic stem cells, right? In the
Are
control
derived
milieu,
from
myself. The embryonic
so
stem cells.
yeah, you can we will discuss that point also tomorrow, but mostly from embryonic stem cells and pluripotent. We prefer pluripotent stem cells. Okay,
Right,
because
yes, yes.
yeah, for because they are, of course, they are not differentiated into a particular function. And because of some ethical issues also, we just use these pluripotent. We can do it with the help of adult also, we can do it with the help of embryonic also, but out of these three we prefer pluripotent stem cells or induced
And ma'am,
pluripotent stem cells.
yeah, induced pluripotent. Because
Yeah.
recently China has, transplanted
Mm.
induced pluripotent developed pancreatic stem cells genetically
Huh.
modified, and
Yes,
the
yes,
patient
yes.
is diabetes free for 2 years. That is a great
Because in
achievement.
case induced pluripotent cell, you don't have to face many kind of issues, and you don't have to have a particular patient to isolate that. You can just
Yeah,
reverse the adult stem cells into a
stem cells
pluripotent
and
stem cell. Yeah, and you
to
can work on that cell. So to avoid all kind of issues and other kind of things, we prefer to use
place.
induced pluripotent cells. Yeah.
But India is not open to all these things. I think still today.
No, not that much. This research is outside India only. But yeah, if we
I've I've
we
faced the
have
music.
discussed
That's
before, as we
why.
have discussed it
Horrible
before.
people sitting there.
Yeah, sorry.
Horrible people sitting there who don't understand anything. And, they just,
Yeah.
keep neglecting your approach for even doing ethical, following ethical guidelines and doing something good for the betterment of the society. They are just bothered about their chair. That's all. I mean, I'm sorry. I
Yeah.
should not be
Yeah.
talking
No,
this,
no, no.
but that is
Exactly. You are hitting a very correct point. But
uh,
yeah, with time maybe we can say some or the other development. We have to see what
the
happens.
government has to set up.
Yeah.
That's it.
Yeah, basically the role is for government only, because scientists are trying their level best to
I had to shut down my stem cell lab because of that.
yeah.
And I mean, it's a waste of money. And I thought that what to do? Let's do the standard procedures anyways. What am I losing? I'm earning anyway, so why do the extra effort? So I stopped my lab, and
Yeah, correct,
we
correct.
again I went back to my basic work. That's all. That's it. Okay, it was
Because
good,
we
good.
are not we are not getting, we are doing lot of efforts, but we are not getting that much of result from from the other party. We are just
Yeah, yeah,
facing
yeah.
some other issues. That's why we we are doing our basic work only. We are
Yeah,
stuck
that is
somewhere.
what the lab work. I mean, all these things I'm doing since last 15 years. I mean, I was
Yeah.
the first person to transplant autologous stem cells in the pancreatic artery,
Oh,
induce the
oh,
results, get them diabetes free for 2 3 years. I have my videos in 2011 on my channel also.
oh,
In
very
in-
nice.
yeah. But then this government are saying what you're doing is crime, and I said, "Oh, nonsense, sir!
Yeah.
I'll rather do my own work." So I stopped everything. I mean, and they were like applying the law retrospective. I said, see, autologous stem cells harvested, given back to the patients. Okay,
Yeah.
after refining. No growth factors added, nothing. It's a pure own stem cells. No,
Oh.
we changed the law.